should be looking at the TCA cycle, which we call the tricarboxylic acid cycle, or the Krebs cycle, or the citric acid cycle. Now, this cycle consists of 8 steps. It's one of the major pathways in the metabolism of carbohydrates, proteins, and fats. Now, acetyl-CoA is the substrate coming in to combine with oxaloacetate at step 1 to give us citrate, involving the addition of a water molecule and the exit of coenzyme A, catalyzed by the enzyme citrate synthase. So what is happening here is simply condensation, the joining together of oxaloacetate and CoA and acetyl-CoA to give us citrate. It's simply a condensation reaction. The top part of this is oxaloacetate, while the bottom part is the acetyl-CoA, just condensation. Step 2 basically consists of 2 steps, sub-steps. The dehydration and hydration, removal of water, and again addition of water to give us isocitrate. So if you compare the structure of citrate and isocitrate, you will see the difference is simply an interchange of groups, side groups. We can see the difference between this and this is that one hydrogen here replace this, and one here replace this at carbon 4 and 5. We see hydrogen coming up here and OH coming up here to compare this and this. So simply a swapping of side groups between this and this, carbon 4 and carbon 5 of citrate gives us isocitrate. And then step 3, we have isocitrate converted to alpha ketoglutarate with the exit of one molecule of carbon 4 oxide. We say it's oxidative decarboxylation. And also there's a removal of 2 hydrogen atoms from isocitrate to give us alpha ketoglutarate. So NAD comes in, collects 2 hydrogen molecules from isocitrate to give us alpha ketoglutarate and also the release of CO2. So if you look at this and this, you will see that the difference is a CO2 group has been lost at this point, and then 2 hydrogen molecules have been removed at this point. So we see that here COOCH2 is CH2 because the COO is gone, so hydrogen replaces it, and then we have CH2 at this point, and then CO, hydrogen here, these 2 hydrogens are removed to give us COO minus. So it's at this point that we have the oxidation, removal of 2 hydrogen molecules at this point, and the exit of the CO2 to give us alpha ketoglutarate step 3, catalyzed by isocitrate dehydrogenase. At step 4, alpha ketoglutarate continues its reaction. Here, CoA comes in, and then another CO2 exits. So at this point, this CO2 exits and is replaced by CoA, pause in here. And also there is dehydration also to give us NADH. If you compare this and this, this and this, there is a dehydration at this point, NADH is formed to give us succinyl-CoA. The CoA came in, and then the CO2 disappears. So this CO2 leaves again, and then we have this at this point. And now step 5, succinyl-CoA is converted. This is catalyzed by alpha ketoglutarate dehydrogenase. Step 5, succinyl-CoA is converted to succinate. If you look at these 2 structures carefully, you will see that the CoA exits. So that here becomes COO, the CoA exits. And then the energy in the thioester bond in this compound, if you refer to our previous lectures on high energy compounds, this is a high energy compound. So the energy in the CoA bond, the bond between succinate and CoA, is used to phosphorylase GDP to give us GTP. And this enzyme is called succinyl-CoA synthetase, named after the reverse reaction in this direction to give us succinate. And then at step 6, we see succinate converted to fumarate. This level of step 5, we say is substrate level phosphorylation. The energy in this bond is used to phosphorylate GDP to GT, so we call it substrate level phosphorylation. At step 6, what we have here is simply dehydrogenation, where 2 hydrogen molecules are removed from succinate to give us fumarate. If you compare these 2 structures, you will see that hydrogen is removed from here and from here to give us this structure. And then the coenzyme responsible involved in this reaction is FAD. So FAD comes in and is reduced to FADH2, and that reaction is catalyzed by succinate dehydrogenase. Step 7, fumarate is hydrated. That is, hydrogen water molecule comes in to hydrate fumarate to give us mannate, hydration reaction. And then we see that here. OH comes here and H comes here, fixes themselves here to give us fumarate, catalyzed by the enzyme to give us mannate, catalyzed by the enzyme fumarase as step 7. And then the last step, since it's an 8-step reaction, is mannate converted to oxaloacetate. Also again, dehydrogenation is taking place. NAD comes in again, collects 2 hydrogen molecules from mannate to give us oxaloacetate. So if you look at these 2 structures very well, you will see that these 2 hydrogen at this point is removed, leaving us with CO at that position. And then oxaloacetate again is free, released again to collect another molecule of acetyl-CoA to repeat the TCA cycle. Now we can see that there are 3 irreversible steps in this entire 8-step cycle. Step 1, 3, and 4 are the only irreversible steps of this pathway. And then we'll see that NADH is produced at 3 steps. Step 3, step 4, and step 8. And FADH2 is produced at step 6. Now remember that 1 molecule of glucose can produce 2 molecules of pyruvate, and then 2 molecules of pyruvate can produce 2 molecules of acetyl-CoA. That means 1 glucose molecule has to go through this step twice to give us 6 NADH, because each cycle produces 3 NADH, 1 FADH2, and 1 GTP. So if 1 glucose enters this, it will go twice to give us 6 NADH, 2 FADH2, and 2 GTP, which can also be used to produce ATP.